What's that? Little bit. It's my pleasure to introduce Michele A. We're lucky to have her come present her work to us. She's an associate professor in the Department of Cell Biology, Neurobiology, and Anatomy at the Medical College of Wisconsin. She got her PhD at Michigan State and did a postdoc with Stephen Duncan, is that right? Also at the Medical College of Wisconsin. She's faculty, or now, like me, she stayed.
That's right.
Michele's work really centers around the transcription factor GATA4, so she is a transcription factor file like myself, and its role in GI development, patterning, and contribution to diseases such as cancer. She sits on a number of study sections, both research and training grants, and she's heavily involved in organizing committees for the AGA. In fact, the things that you see at DDW is because of Michele and decisions that she makes in these committees. She has really a passion for mentoring, and if you've ever seen one of her students give a talk at any of the meetings we go to, you will know what I'm talking about. She really is an outstanding mentor. It's a pleasure to have you out here, Michele. We're looking forward to your presentation.
Thanks. Is there a way to get that?
Yeah, that's what I was just saw.
Sometimes when there's chat and something.
No, I chatted to make sure.
Oh, okay. There we go. All right. Okay, well, it truly is my pleasure to be here today. I'm very excited. I've never actually been to this area of the country before, so it's really been great to be here, and I've had a fantastic visit so far. Everyone is so welcoming, so friendly. I've learned so many great research things. I'm just really thrilled to be here. So thank you for being here now and listening to what I have to say about the work I've been doing for the last, I guess, 15 years in my lab and 5 years as a postdoc at MCW. Let's see, wrong thing. Okay. Advancing? No. Like this. I tried the down arrow. Okay, I'm still not skilled at this, right? Okay. So my lab at MCW is really focused on studying epithelial tissue development and looking at diseases through this window of developmental biology. And so what we think and what we believe and what we work upon the idea is that the processes that happen to form an organ, often these developmental processes can become abnormally activated in diseases. And so if we understand those fundamental mechanisms that make the epithelium of the esophagus or the stomach or the small intestine, then we believe, is that showing up for you all? You don't see the pointer, okay. So everyone on Zoom sees it, but you all don't see it? Okay. That's okay, I can point to this too. But so get back to where I was. So if we understand those basic processes, then we can look at things going awry in disease and apply those ideas to that. So can we correct things in disease because we know how it should happen, right? And so I'm not like the smartest person in the world, because the first thing I thought of was, well, how does this happen? Okay, we have the endoderm. It's divided into foregut, midgut, hindgut, and now from there we make an esophagus, a stomach, a small intestine, a colon. Well, how does that happen? Well, clearly transcription factors, right? So it's not like a mind-blowing idea that the transcription factors that are expressed throughout the endoderm are going to be responsible for turning on the genes to express the proteins that will make, for example, a parietal cell in the stomach to secrete acid, or an enterocyte in the intestine to absorb nutrients. And really, it's not a single transcription factor, right? It's the combination of factors together that are going to lead to an organ's specification and cell type differentiation, and the morphogenesis that allows for the correct architecture of that epithelial tissue. And so there may be factors that are ubiquitously expressed. There may be factors that are expressed in gradients, factors that are uniquely expressed in certain areas of the endoderm. But taken together, these transcription factors program the endoderm to take on these organ identities, to make a multi-layered stratified squamous epithelium in the esophagus, which is important for the function of the esophagus, right? It needs to be a protective epithelium to withstand the forces of friction and the injuries, the chemical injuries, the hot temperature of beverages you take in. But of course, in the small intestine, for example, you need those absorptive cells. You need to have villi so that you can have increased surface area for absorption. Or in the stomach, you need chief cells to secrete digestive enzymes, and you need parietal cells to secrete acid. So really, I got interested in the GI tract in wanting to understand how transcription factors work to control this morphogenesis and then cell specification and differentiation and patterning. And in terms of diseases, of course, we study the GI tract because we know that GI diseases are a big problem. They cost a lot of money. We have Barrett's esophagus, we have esophageal cancer, gastric cancer, IBD, short bowel syndrome. So right now, we're focusing mainly, and today what I'm going to talk to you about is the work we've been looking at to understand how we can apply developmental biology findings to the metaplasia that happens in the esophagus in Barrett's esophagus, which is a columnar metaplasia that is the only known risk factor for esophageal cancer. And so what does Barrett's look like? So what happens in Barrett's esophagus is that this normal multi-layered stratified squamous epithelium undergoes a transformation to this simple columnar-like epithelium that takes on the character of gastric and intestinal cell types. And in and of itself, Barrett's metaplasia is not a problem. But as I said, some people who develop Barrett's metaplasia go on to develop esophageal adenocarcinoma. And esophageal adenocarcinoma is a very devastating disease. In general, esophageal cancer is the 7th leading cause of deaths worldwide. 1 in 20 cancer deaths worldwide come from esophageal cancer, and adenocarcinoma is the fastest increasing cancer in Western countries. It is often not diagnosed until very late stages, and so when esophageal cancer is diagnosed at late stages, there's only about a 5-year survival rate of 5%. When it is diagnosed earlier, you can have survival rates, although not great, 40 to 50%. And so we believe that understanding these processes that give rise to the early stages. So how does this squamous, what happens in this tissue in the context of injury? So in reflux, so GERD is a risk factor for this. How does this tissue transformation happen? Can we understand how it happens so that we might be able to reverse it? Can we understand how it happens so that we might find new ways to screen for it other than invasive endoscopy? And can we understand how this moves to cancer so that we can make inroads into perhaps identifying cancer earlier or finding better treatments? And so, can understanding the mechanisms of early esophageal development provide us clues to the pathogenesis of Barrett's esophagus? And so, during development, the esophageal epithelium, again, comes from the foregut endoderm. And this foregut endoderm has sort of a simple columnar morphology, and it actually expresses the transcription factor of interest that I love, GATA4. But as development happens and this turns into a stratified squamous epithelium, GATA4 is lost. Now in disease, we see an opposite effect, right? A healthy esophagus is a stratified squamous epithelium. In the context of reflux and inflammation, a metaplasia happens to convert this to a columnar metaplasia, a columnar epithelium. And so really the question is this really sort of development reactivated and gone backwards? Have we taken this stratified epithelium and moved it backwards through some developmental pathways that have initially taken this columnar epithelium to squamous? And if that's the case, then can we actually use this knowledge to reverse this? And as I mentioned, transcription factors are my bread and butter, and GATA4, which is a zinc finger transcription factor, is the one that we've been working on the most. And so it is expressed, as I said, in the foregut endoderm, and it's turned off as the epithelium of the esophagus becomes squamous. And I should mention in the mouse, where we study this in the mouse, and the way we study the esophageal development versus stomach development is in the stomach, where we have a forestomach and a hindstomach. So the mouse has this little extra bit of tissue known as the forestomach, but is pretty much analogous to the esophagus. So this junction between the squamous epithelium, see it better here, and this developing columnar epithelium in this 18 and a half, this embryonic stomach from an 18 and a half day old embryo, we can basically call this forestomach, it's the esophagus. This is a squamocolumnar junction. So you can see GATA4 staining here in this hindstomach epithelium that is taking on this glandular morphology, and it's absent in the forestomach. This is just another way to look at this. This is E-cadherin staining, which I think nicely shows the squamous epithelium. And we've stained with GATA4 in red. And so you can see the GATA4 positive nuclei populating the columnar epithelium, where you see the lack of GATA4 in the squamous epithelium. So the next question was, if we had this idea that turning off GATA4 is important to make a squamous epithelium. In this disease, would GATA4 be expressed in this columnar epithelium? And so we were able to obtain tissue samples and look at that. And so just like in the mouse, the normal mature stratified epithelium of the esophagus or the forestomach lacks GATA4. The same is true in the human. So here's a section of human esophagus, we lack GATA4 protein. But in disease, Barrett's, and in cancer that arises from Barrett's, GATA4 protein is present. So if in contrast to development, where we see GATA4 turning down to make a squamous epithelium, now we see reactivation of GATA here when this squamous epithelium undergoes metaplasia to a columnar epithelium. So we had this simple idea that GATA4 was an important signal here. And so that this foregut endoderm that starts out expressing GATA4 then becomes programmed and patterned such that GATA4 is repressed or turned off, and that happens and allows the stratified squamous epithelium to develop. Whereas GATA4 is maintained in the region of foregut that gives rise to the hindstomach, and simple columnar epithelium develops. And so we ask these questions basically: is GATA4 necessary to form a columnar epithelium? And is it sufficient to convert a stratified epithelium into a columnar epithelium? And the work I'm going to tell you about now was recently published in CMGH. A very talented postdoc, Annie De La Forest, and a previous graduate student, Bridget Kohlenhofer, were really the drivers of this. And so we were able to use some mouse models we had made. We were able to use a conditional knockout model of GATA4 in order to ask, is GATA4 necessary? So we were able to turn off GATA4 in this endoderm using sonic hedgehog Cree. And what we postulated was that when we turn GATA4 off in this endoderm, instead of forming a simple columnar epithelium, a stratified squamous epithelium would emerge. On the other hand, we had a nice mouse model that I made at the end of my postdoc, where we put the GATA4 coding sequence in the ROSA 26 locus as a conditional knock-in. And so again, using sonic Cree, we could turn on GATA4 in this endoderm and ask the question, if GATA4 stays on or is maintained, will that be enough? Is that sufficient to convert this epithelium that should be squamous into columnar epithelium? And so first looking at the knockout situation. Indeed, we saw that when we knocked out GATA4 in the hindstomach, the epithelium took on a stratified squamous morphology. So here in this section of a day 18 and a half embryo, mouse stomach, you can see this nice glandular simple columnar epithelium emerging. Again, E-cadherin is in green and GATA is in red. However, in this mutant, where GATA4 is absent, you can see that this epithelium doesn't have this simple columnar glandular morphology. It has a stratified squamous morphology. Interestingly, there were regions in the mutants that maintained GATA4 expression. So the Cree wasn't completely penetrant, and we didn't get complete recombination. In those regions where GATA4 was maintained, we actually saw that those areas looked more like the control and were developing more of a glandular type structure, suggesting that this phenotype was really a cell autonomous phenotype, that the loss of GATA4 in this tissue was responsible for this stratified squamous morphology to occur. So at this point, we saw that we had a structural phenotype, a change in the architecture. The question now was, is it still programmed as a columnar epithelium? Are there still gastric cell types there? Or have we actually changed it at the identity level as well? Do we just have a stratified epithelium that will have parietal cell markers and chief cell markers, or are we losing that? And so we did some staining, and we saw that when we stained for a parietal cell marker, ATP4A, or for a chief cell marker, GIF, we saw that in the day 18 and a half embryonic stomach of controls, you could see these brown stained cells here. Those are ATP4 positive parietal cell progenitor cells, or new progenitor, or newly forming parietal cells. And you could see GIF staining as well in these emerging gland bases that were forming in the embryonic stomachs of the controls. However, in these regions of stratified squamous epithelium that were lacking GATA4, you saw a complete absence of these markers, suggesting that it's not simply we're changing this tissue architecture, but we're changing the tissue identity and the cellular identity. So we're losing differentiated cell populations when we eliminate GATA4 in the developing columnar epithelium. You might predict then, or ask the question, well, if you are seeing the loss of columnar morphology, you're seeing the loss of these cell types, do you see the gain of squamous markers? And so that's exactly what we saw. We looked now at staining for some stereotypical squamous epithelium cytokeratins. keratin 5, keratin 13, keratin 14, and we saw that we saw that when you lost GATA4 and you had this stratified squamous looking epithelium, you now saw these markers of squamous epithelium turning on. These are present in the control forestomach as you'd expect, but absent completely in the hindstomach as you'd expect. And interestingly, this key transcription factor, P63, which had been shown to be a master regulator of stratified squamous epithelial cell development, was highly expressed in the tissue lacking GATA4, similarly to the control forestomach. And it was completely absent, of course, in the hindstomach. So at this point, we thought loss of GATA4 in the developing mouse stomach led to a change in the cell architecture. So we had a squamous epithelium, and led to a loss of the expected differentiated cell types and a gain of cell types that you'd expect in the esophagus, and the activation of this key transcription factor P63. Now, all the data I've shown you so far is at day 18 and a half. So we decided to step back a little because, of course, we know sonic hedgehog cree is turning on much earlier, and we wanted to really get at whether this was a primary effect or this was some sort of secondary effect happening. We're looking, 8 days later to when we've turned off cree. So we went back and we looked to see, using a careful morphological and histological analysis, at what point we could start to see the loss of GATA4, and also look at that at the time at which the epithelium would begin this transition from foregut endoderm into a stratified squamous epithelium or a columnar epithelium. And it turns out that at day 13 or 14 and a half was the time point that we should look, because that is the first time point we could start to see this morphogenesis happening. And also it was, because of the activity of sonic cree in the hindstomach, it's not as fantastically penetrant as, say, like villain cree or something like that. So it took a little bit of time to actually get a sufficient knockout of number of cells expressing GATA4. And you saw that we never really get rid of all of them, but we get rid of most of them by that time. And so we looked to see what the phenotype looked like in day 14 and a half embryonic stomachs. And we saw that when we looked at these markers, again, by qPCR this time, of cell types like the surface mucus producing cells or the parietal cells, we saw that these markers were all lost in the GATA4 knockout. So these are epithelial cell preps from day 14 and a half, control or conditional knockout stomachs. And all of these markers were lost, showing that even at 14 and a half, at this time point in which the morphogenesis is beginning to happen to say, this region of endoderm is a simple columnar epithelium, expresses GATA4. This region has turned off GATA4 and now will become stratified. When we've lost GATA4, we've lost the emergence of these differentiated cell types. And we see that these markers of squamous epithelium are turned on. Particularly, we can see P63. This is to compare to normal control forestomach here. And you can see that P63, keratin 17, and keratin 15 to some extent are all induced. So at this point, I hope you believe me when I say that GATA4 is necessary to drive columnar epithelial morphogenesis. When we lose it, we convert the tissue to squamous and we lose differentiated cell types and we start to see esophageal cell types emerge. So now on to the next question, which is it sufficient? So going now and turning to this knock-in model, I'm now going to show you data from when we turned on GATA4 using sonic cree. So we didn't allow GATA4 to be silenced, and look to see what's going on in this forestomach. Are we going to make a simple columnar forestomach? And that's exactly what we saw. So these are again at day 14 and a half. So we now are focusing at day 14 and a half. And what you can see here is in this H&E of the controls, you can start to see, and I think it's pretty clear here in this IHC here, a multi-layered stratified epithelium beginning to emerge. And of course, it doesn't express GATA4, and it expresses keratin 5, which is a key marker of squamous epithelium. When we now have GATA4 expressed in this tissue, you can see that this tissue looks much more like a columnar epithelium, and we see a complete loss of keratin 5. Suggesting again that when you abnormally activate or topically express GATA4 in that tissue, where it should be silenced to drive squamous epithelium morphogenesis, you can convert that tissue to a simple columnar epithelium. And so given that GATA4 is a transcription factor, we wanted to look at this more globally. Right now, I've just picked out a couple markers here and there. You might say, well, sure, you see a couple markers change, but what does this look like on the whole? What does this look like globally in the transcriptome? And so we took an RNA-seq approach. And so the first thing we did was that this is bulk RNA-seq. We first took the control hind stomach epithelium and the control forestomach epithelium and compared those because we wanted to establish a benchmarking set of genes. So the set of genes that was highly expressed in forestomach to benchmark the identity of forestomach epithelium, and a set of genes that was highly expressed in hind stomach to benchmark the hind stomach cell identity. So now we could take these sort of benchmark sets, perform RNA-seq on our mutants compared to our controls, and look to see what was happening at a global level. Were we actually seeing induction of this forestomach pattern at a more global level in the knockout hind stomachs? And were we seeing induction of this hind stomach pattern in those forestomachs that now had GATA4? And so first looking at the conditional knockout, so this is the GATA4 deficient hind stomach. And what we saw that was among the sets of genes that were changed that mapped to this hind stomach gene expression set, we saw that we had about 500 and, I should have done the math there, 544 genes changed, and about 1,250 or so genes changed in the 4 sets gene expression set. Now, we would predict that because we are eliminating GATA4 and getting a squamous identity, we should see that the hind stomach genes are downregulated and the forestomach genes are upregulated, and that's what we see. The vast majority of these genes that would mark hind stomach are downregulated when you eliminate GATA4, and those that mark forestomach are upregulated. We then used some ChIP-seq data we had to look to see whether or not GATA4 had binding sites in these genes. And so it was really impressive, I think, when we looked at these genes, these 519 genes that were downregulated, when GATA4 was lost in the hind stomach, 72% of those had GATA4 binding within the plus or minus 2 kb of the transcriptional start site. And 56% of those forestomach enriched genes also had GATA4 binding sites, suggesting that this is not only just a consequence of GATA4, but an actual direct consequence of GATA4 loss. So that GATA4 would normally be binding to these genes that induce hind stomach identity and positively regulating this gene expression, and actually, really interestingly, binding to the genes that should be repressed. So binding to those genes that encode forestomach identity and repressing those, and thereby promoting columnar cell fate. On the knock-in side, we saw a very similar effect. So in this case, because we're putting GATA4 in the squamous epithelium, we see that simple columnar epithelium emerging. We expect that those forestomach genes should be downregulated, and we should see columnar hind stomach genes upregulated. And that's what we see. We see that the vast majority of genes that map to this benchmarking hind stomach gene set are upregulated when we have GATA now in that squamous domain, and those that mark this forestomach gene expression set are downregulated. And again, the vast majority of these are bound by GATA4, again suggesting that GATA4 is directly driving this phenotype. So it is directly activating genes that should drive the hind stomach pattern and repressing genes that would drive a forestomach pattern. So we now wanted to look at this mechanism a little more deeply. And so we know that GATA4 is directly regulating a large set of genes that actually confer that identity. For example, like keratins, we looked at that, or like ATP4A, for example. But we also had this really nice data set that came from Ramesh Shivdasani's lab that had identified a set of transcription factors that were esophageal specific and were really important in development of esophagus stratified squamous epithelium. And we looked at those factors and we found that in total there were about 15 or so in the paper, and we found that about 2/3 of those had GATA4 binding sites. So I'm showing you here the data of those transcription factors that we think GATA4 would bind to. And we saw that those transcription factors that were normally not expressed, of course, in columnar epithelium, the vast majority of those were turned on now when you lost GATA4. On the other side, when you normally should express those factors, because this is an esophageal type, a squamous epithelium, now when you added GATA4 in, you saw these were being inhibited and these weren't expressed. So this data suggested to us that this network of transcription factors that was really essential for programming stratified squamous cell fate was being repressed by GATA4. So these all are bound by GATA4, and it suggests that GATA4 not only promotes columnar cell fate by driving expression of those genes and proteins you need to make a columnar cell, and by repressing the genes and proteins you need to make a forestomach cell, but also it enforces that by regulating this transcription factor network, by repressing this network of transcription factors required to make a stratified squamous epithelium. So getting back to disease. So I said in the beginning, I wanted to study development as a window to disease, and I mentioned that Barrett's esophagus is a model that we think we see. Now, when we see metaplasia, when we can convert a stratified squamous epithelium to a columnar epithelium, it looks a lot like Barrett's esophagus. We see that metaplasia. So if we take our gene set from our genes that are changed in our knockouts and in our knock-ins, how does that align with data from disease? So there had been some papers that were published using RNA-seq to identify gene sets of interest in Barrett's esophagus. And so we took that set of genes and now said, is there any overlap? Does what we're doing in development look at all like disease? And so when we looked at the gene sets that were upregulated, so transcripts that were upregulated in Barrett's esophagus, we saw that those genes were downregulated when you lost GATA4. So these are columnar type transcripts. So you'd expect these to be lost when you lose columnar identity. And those that are upregulated let's see here, sorry, I confused you. These are the upregulated in Barrett's, of course, are downregulated. And on the other side of the coin, now this is more like the disease state. This is our knock-in model. So this is when we're adding GATA4 in and we're seeing that we have a squamous epithelium converting to columnar. You see that these genes are highly induced. So these genes that mark columnar epithelium and that are indicative of Barrett's esophagus are being driven in our mutants. On the other hand, the genes that are downregulated, so those that would be associated with more a squamous epithelium, those are upregulated when in the hindstomac lacking GATA4 and downregulated in the forestomachs that express GATA4. And I don't have the exact percentage here. Sorry, I should have written it down, but the vast majority of the genes shown here also are bound by GATA4. Again, suggesting it's not simply an indirect effect, but the GATA actually directly binds and regulates this gene expression. So, taken together, I hope that I've convinced you that GATA4 regulates epithelial morphogenesis in the developing stomach to promote columnar versus squamous cell patterning. And so what we think is going on in this, summarized in this cartoon, is that when the endoderm turns off GATA4, that allows for forestomach gene expression to happen and for emergence of squamous epithelium. And when GATA4 stays expressed in hindstomac, of course, that allows for the simple columnar epithelial program to be put in place and for columnar epithelium of the hindstomac to emerge. So we think that in this tissue that expresses GATA4, GATA4 is acting in multiple ways. GATA4 is repressing those genes that define forestomach. Not only the genes like the cytokeratins, but a network of transcription factors as well. And it is positively regulating the network of genes that turn on hindstomac simple columnar epithelial cell fate. Now, when we've eliminated it and a squamous epithelium emerges, we see that we've lost this ability. We've lost GATA4, so we can't now repress this forestomach gene fate. And so now this network of transcription factors is activated and squamous cell identity happens. And of course, the hindstomac genes are not expressed. And on the other hand, when we add it now and we put it into the forestomach, we now allow GATA4 to do what it would normally do in a hindstomac, but now in the forestomach. So it represses those forestomach genes, represses that network of transcription factors, and activates the hindstomac gene expression pattern. And in that way, converts this tissue to a squamous epithelium. So back to disease. So this is all great. We see this in development, right? So development, of course, has a lot of plasticity. That endoderm tissue, the developing tissue, there's a lot of plasticity there. So the question is now when we go to disease, like would these same things be true? Would you see that if you looked in an adult stomach or you looked in a mouse stomach where you can induce disease, would you see the role of GATA4 would GATA4 be playing an important role in disease? So we looked to find a way that we could model Barrett's esophagus in mice. And it's not a simple procedure, but you can use a surgery, an esophageal gynostomy surgery, to allow reflux to happen in the mouse esophagus. And in the conditions of reflux, in normal control mice, metaplasia can happen, not in all the mice, but in maybe about 20% to 30% of the mice, metaplasia happens. Dysplasia sometimes, but never cancer. And so we asked the question, if we use this model where we're priming the cells with GATA4, so now we're using our knock-in model, we've switched Cres here, we're using EDL2 Cre for two reasons. One, EDL2 Cre is very specific, so it's expressed in forestomach, esophagus, and skin, whereas of course sonic hedgehog Cre has much broader expression patterns. And actually, because our knock-in animals did not survive past birth, they had neural tube defects, likely because sonic was inducing GATA in the developing neural tube where GATA shouldn't be. We couldn't use those animals to move forward. So we switched to this EDL2 Cre model. And what we thought was, if we put GATA4 in this tissue and now do this surgery where we join the esophagus to the jejunum, what would we see? Would we see just no effect? Simply you see stratified squamous epithelium and GATA4 has no effect. Would we see that maybe Barrett's would be advanced? So would we see Barrett's in more percentage of the mice? Would we see Barrett's sooner? Would we see larger areas of Barrett's? Would we see cancer? Would it possibly accelerate the progression of Barrett's to cancer? And so I probably wouldn't be showing you this unless there was something interesting and exciting to see, right? And so what we saw was that in all of the mice that were the GATA4 conditional knock-in animals, so all of the mice that had GATA4 in the esophageal epithelium, every single mouse we've looked at, starting at 3 months on, has developed cancer, has developed tumors. And so that was really shocking and striking to us. Those tumors, this is an example of one that came from an 11-month mouse, are quite invasive. So you can see that this tumor invades all the way into the muscularis. You can see that the cells express GATA4. You can also, I think, appreciate from this that, especially if you are a pathologist out there, that this is not clearly an adenocarcinoma as we would have predicted. So our prediction was, of course, that GATA4 drives metaplasia and then metaplasia leads to cancer. This actually had a lot of squamous identity. A lot of when we had a pathologist look at it, they said that, this is more of an adenosquamous cancer. And so we were a little bit surprised by that because we didn't expect GATA4 to drive squamous cell cancer. And of course, reflux and GERD, that's not really associated with squamous cell cancer either. And we didn't know if GATA was even expressed in patients in squamous cell cancer. So we wanted to say, okay, let's step back a minute now. Is GATA4 relevant in squamous cell cancer? Or is this, is our model going to be something that we can use to look at human disease? Or is this just something about a mouse and giving it a reflux surgery? You see this happen. And so we did obtain some samples of squamous cell cancer, and we did find that GATA4 was expressed not only in adenocarcinoma, but also in squamous cell carcinoma. There are also tumors out there that are adenosquamous that happen in people, and we had a couple of those. They're very rare, but we did see GATA4 expressed in those as well. I just don't have an image to show. I will say, interestingly though, although all of the adenocarcinomas we've ever looked at express GATA4, that's not the same for squamous cell cancer. There are a subset of tumors that we looked at that don't express GATA4. There's also variation in the intensity of GATA4. So some tumors seem to express a lot of GATA4 protein, some a little. Some have 5% positive cells, some have 95% positive cells. And so it's very different from adenocarcinoma in that way that there seems to be heterogeneity about GATA4 and squamous cell cancer. And so right now we're trying to work on understanding that. Are there any connections there to say the stage of the tumor, the grade of the tumor, possibly any characteristics of the patient's obesity, smoking, alcohol use? We're trying to understand why that's happening. Is there something clinically relevant there that we can dig into? So that's a really exciting thing we're doing. Of course, we need more squamous cell tumors, and we're working with a colleague now at the tissue bank at MCW to get those samples, to stain them, and a medical student is able to go back to the patient histories and try to tease this kind of stuff out. Now we are really interested in our tumors in the mouse though. Now that we have a little more confidence that what we're seeing may actually be important to human disease, because we do see that GATA4 is expressed in both adenocarcinoma and squamous cell cancer. And so we took a spatial transcriptomics approach to now look at what's going on in these tumors. We wanted to take the spatial approach because we knew that, for example, our tumors are very heterogeneous. So if we just grind up the tumor and do an RNA-seq, we're not going to be able to understand that heterogeneity. And even if we do a single cell seq, we can't really understand how the cells are acting in relationship to each other. We can understand all of the various cell populations we have, and I think it's a very important experiment and we want to do it. We just haven't had animals to do it. But we can now with spatial transcriptomics try to understand what's going on regionally. So we can identify areas that look like squamous cell cancer and understand what's going on there. We can look at regions that look like adenocarcinoma. We can look at normal regions, and we can get an idea of what's going on in that environment. What sort of immune cells are there? What sort of mesenchymal cells are there? And really try to really get to understand molecularly what's going on here, and then take that back to the human tissue. And so we performed spatial transcriptomics on tumors from 4 mice at 5 months old. I don't have a ton of data to show from it because it's really a relatively new experiment from the lab. And I will confess, bioinformatics is not our strength. And so we have been working on this. It's not simple data to analyze, but Olivia Franklin, a graduate student in my lab, has been working really hard on this. And so if anyone out there is a spatial transcriptomics expert or has interest, bioinformatics person, contact me. I would love to work with you because this is not something that we are experts on. Talking to this lab, right? But one thing we were able to do pretty simply, just to sort of get an idea of whether or not what we could find from it. The first thing Olivia did, just very simply across all of her tumors, was she circled regions or lassoed regions of tumor. She didn't actually break it down from squamous regions or adeno regions yet, and regions that look normal. And we said, well, what do those look like? Can we at least see that there's cancer and one is normal? And so when she did some gene set enrichment analysis, she was happy to see that. When she looked at the cells, the lassoed regions from tumor, she saw enrichment for pathways in cancer. She saw enrichment for transcriptional regulation in cancer. So the good news was, at least from our first level analysis, which is not, it's pretty superficial, admittedly, but we could at least look at this and say, okay, we can now look at some tumor cells and regions of tumor across our animals and look at some regions of non-tumor. And we looked just quickly to see what kind of pathways were turning up, and we were excited to see that some interesting pathways that were positively correlated with the GATA4 tumor cells versus the GATA4, and these are GATA4 positive non-tumor cells. So we saw that signaling receptor interactions between extracellular matrix and that were upregulated. This could be interesting because we see invasive tumors, right? So we see these tumors invading. We saw PI3 kinase and AKT signaling and TGF-beta signaling positively correlated. And so right now what Olivia is doing is, one, digging into this a bit, and then two, really working hard to take this to a deeper level of understanding. Because I really do think we have a fantastic data set there that's really going to help us make inroads to understanding squamous cancer and adenocarcinoma, and give us a really exciting data set to go back to in our patient tumors and look at, and then hopefully be able to do this in patient samples. And that segues well into the next thing that I wanted to talk about that's going on in the lab, and that is actually going back to human samples. And so we are fortunate to be working with Ramon Jin, who's at Baylor College of Medicine, and he's a physician scientist who has been able to start to develop a bank of human esophageal organoids across a spectrum of disease, from normal to Barrett's to cancer, that we can now use to try to, one, look at the role of GATA4 in that, and two, anything we find in our animal model, now go back and test this in an organoid model. And so this is just to show a couple of organoids that he's taken here from biopsies. This is the biopsy H&E. This is an organoid H&E. You can see that the simple columnar epithelium that you see in the biopsy is present in the organoids. And also, one important marker of Barrett's esophagus is mucin 2 production, and Alcy in blue is showing you that you're seeing that in these organoids. So these organoids are really representing well, at least structurally and phenotypically, what you see in the biopsies from the patients. So the next question was, do they express GATA4? And we found that they do. Hope this is coming through well enough here. Maybe you can see it best here. You can see this brown nuclear staining here. This is GATA4 protein staining on these organoids. And you can see, I think, that there is a range, though, in these organoids. So some organoids seem to express a lot of GATA4 protein, whereas some express a very low level. That was actually held up in the RNA levels as well. These dots here, this U15, 16, and 12, those are the samples I'm showing you here. And you can see that 16, which has the highest RNA level, also has the highest protein level. And number 12, that has the lowest RNA level, has the lowest protein level. And these are some other Barrett's organoids. So you can see there's a range of GATA4 in these organoids, which can be very interesting because we haven't really looked at so Barrett's can be broken down more than just Barrett's esophagus. You can look into Barrett's with intestinal metaplasia or gastric metaplasia. There's some debate in the field whether really it's only Barrett's with intestinal metaplasia that matters for disease. If you're in the US, you might say that. If you're in the UK, you wouldn't say that. And so there's a lot of debate about that. No, it's true because the doctors need to know, right? When we're doing endoscopies on patients with Barrett's, we have to follow up on these people, but who do we follow up on? Do we give everybody endoscopies all the time? Or if it's really like intestinal metaplasia, that's the marker. So then we find the patients with intestinal metaplasia and we follow up with them. But if gastric metaplasia can be just as important in disease, but you're missing that, then you're missing that group of patients that may then go on to develop very advanced and terrible esophageal cancer. And so I think this is a very interesting model where we might be able to look at different levels of GATA4 and how they are contributing to that Barrett's esophagus phenotype. We also have organoids from cancer. So these are two patient samples from adenocarcinoma, and you can see that GATA4 is expressed in these organoids. And so far we have one from squamous cell cancer, and fortunately it happened to be from a squamous cell cancer that expressed GATA4. So we're building this organoid bank to try to then use that as a resource, as a tool to now study in human disease what we learned in the mouse. We've done one preliminary experiment that I was pretty excited about with Barrett's organoids, and it was a GATA4 cut and tag. So we wanted to look to see in the Barrett's organoids, in the Barrett's tissue, what genes is GATA4 directly binding to, and what are they? Are they important? Are they really exciting? Can we get excited about them and like have some great clues to get working on and studying? And I feel like every time you give a talk, I wouldn't say that unless something interesting was coming out, but yes. So the cut and tag showed us some really interesting, promising candidates. So of course, we were interested in seeing that GATA regulates itself. So GATA was bound to itself. It was also bound to GATA6, which is a family member that has often been shown to have functions that overlap with GATA4. So GATA6 is expressed and GATA bound to it. We saw CDX2, which is a really key transcription factor that defines intestinal cell types, defines intestinal morphogenesis. It's also one of the key markers that folks look for in Barrett's esophagus. We saw one of this group's favorite factors, SOX9. SOX9 was having GATA4 bound to it. We saw beta-catenin, so Wnt target, keratin 8, which is a key cytokeratin of columnar epithelium, and then DDIT4 and IFRD1, which I don't know if you follow Jason Mills's work in paleogenesis. Did I say it right? Palaginosis, thank you. Palaginosis. These were two really important key molecules that they had implicated in the process. Thank you, Joseph, for the help. Pronunciation isn't my best suit. So this was pretty exciting to us, and so we're following up on these now. And some of the great things you can, of course, do with organoids is you can CRISPR knock things out, you can do double knockouts, you can overexpress things, and so we're working on this to try to understand if we modulate and manipulate these things, how does that affect that phenotype of Barrett's esophagus? So in the last 5 minutes, for Joseph, I was talking to him last night about how we're now taking a foray back into the stomach. So our developmental biology studies of the stomach led us a bit to the esophagus and Barrett's esophagus and cancer, but we're going back to the stomach now, and we're interested in how GATA4 functions in the mature stomach, and we want to define the role of GATA4 specifically in parietal cells. And so why study gastric parietal cells? So parietal cells are the acid-producing cells. And what we know from much work, particularly from Jason Mills's lab, that when you lose parietal cells, so when there is parietal cell loss, that drives a process of metaplasia. And loss of parietal cells is an absolute requirement for that process. So parietal cells must be lost as one of the first steps, some would say, in the process of gastritis and metaplasia and then cancer. And so when the parietal cell is lost, that allows for metaplasia to happen. The chief cell can undergo a dedifferentiation, form a metaplastic cell, and then in this prolonged state of metaplasia, cancer can arise. So the parietal cell is a really key cell. If we could understand how that parietal cell was developing, how it was maintained, how do you maintain a parietal cell, and can you then regenerate a parietal cell possibly and understand ways to stimulate parietal cells, that could be really important in understanding and blocking or perhaps preventing or fixing perhaps patients who undergo have this chronic atrophic gastritis, loss of parietal cells, and maybe then intervene to prevent metaplasia and cancer. And so we thought that GATA4 was important based on a couple pieces of data. We saw that one, GATA4 is expressed in parietal cells, so of course that's important, right? No reason to study GATA4 if it's not there. So this is an immunofluorescence showing the ATP4A marker, which is part of the proton pump stained in green, and GATA4 stained in red. And so these parietal cells nicely express GATA4. This is just an immunohistochemistry showing the same thing and also showing that in the mouse, GATA4 is also expressed. So this is human and this is the mouse, showing that GATA4 is expressed in parietal cells in both the mouse and human. We want to use the mouse as a model, so we wanted to be sure, of course, it's expressed in the mouse parietal cells. There was also a paper from back in 1993 where they had done some EMSA analysis, binding, in vitro binding, and they found that GATA factors could bind to the promoters of the acid pumps of ATP4A and 4B, suggesting GATA might have a role in regulating some really key molecules in parietal cell function. Jason Mills's group had done some work where they had sorted out parietal cells and done some Affymetrix gene arrays. The young people in the audience will have no idea what I'm speaking of, but the old-fashioned way to look at transcriptomics. And so what he did was he defined from that a set of about 230 or so genes that had expression that you would call enriched in parietal cells. And when we looked at our ChIP-seq data from mouse stomach, we saw that 81% of that set had GATA4 binding sites, having GATA4 bound, so ChIP-seq peaks from GATA4 pull down. 73% of those had binding in what you would define as an enhancer and a promoter, and there was a small subset that had either a promoter or enhancer binding set. But 81%, so the vast majority of these genes that would define a functional parietal cell seem to be bound by GATA4. So taken together, we thought that GATA4 seems like a really important transcription factor that may be regulating either the development of parietal cells or the function of parietal cells. So since we have the knockout and the knock-in tools, why don't we now investigate GATA4 in these cells? So we were able to get an ATP4B creed, which would allow us to knock GATA4 out in those parietal cells uniquely, not in other cells of the stomach, but just the parietal cells. And we asked this question: if we are to eliminate GATA4 in these parietal cells, which I've drawn here in green, will the loss of GATA4, will it lead to this, what you see when you lose parietal cells? For example, if you use tamoxifen to damage the stomach, you'll see a loss of parietal cells, and you'll see these features of metaplasia. You'll see hyperplasia. You'll see loss of parietal cells, and you'll see stem cells coming out. So these chief cells transdifferentiating into these metaplastic type cells. So we used our conditional knockout again with ATP4B creed, and we looked to see what happened. In our animals that expressed lost GATA4 in the parietal cells, you can see that there is a nice loss of GATA4. So these are parietal cells lacking GATA4. But you might have heard what I just said. These are parietal cells lacking GATA4. So they still express this ATP4A marker, but they looked a little bit different. So we were intrigued. They were a little bit smaller than these control cells. They had overall a lower cytoplasmic to nuclear ratio. So when you look at a parietal cell normally, it has like this fried egg morphology. So you see this nice cytoplasm and central nucleus. And in this case, our cells were a little bit abnormally shaped, and they seemed, for the most part, not all, to have a lower cytoplasmic to nuclear ratio. Functionally, we saw that the glands in the stomachs lacking GATA4 underwent this hypertrophy. So the glands were much longer in the stomachs that you saw lacking GATA4 in parietal cells, which is, again, a marker of SPEM. We saw that proliferation was increased. So this is an EDU. So we pulsed with EDU and stained for cell proliferation. And you can see here that we have an increased number of EDU positive cells. This is a zoom in from here. If you compare those animals lacking GATA4 in the parietal cells to those expressing GATA4 in the parietal cells. So when we counted these, we saw that we had, in general, an average of, we had more epithelial cells per gland, as you'd expect. We had more proliferative cells per gland. And when we looked at the pH was elevated. So it was less acidic. So even though parietal cells, at least morphologically, appear to be there, they're not functional. And this actually suggests that you don't necessarily have to lose the parietal cell completely to drive this. You have to lose the function of the parietal cell. So the acid-producing function and perhaps maybe some of the other products that parietal cells make to help maintain the environment of the other epithelial cell types, the other important gastric epithelial cell types. We did some staining to look for SPEM markers, and so that's the metaplasia. And we did see that you shouldn't really see co-staining for GIF and GS2. These are neck cell and chief cell markers. And so these regions of yellow are showing you cells that are co-expressing these markers and are indicative of metaplasia, of SPEM cells. But we also noticed something else really weird. We saw that these cells that looked like they were parietal cells had GIF expression. So that's a chief cell marker. You shouldn't expect to see this in the parietal cells. So we said, well, okay, maybe this is just some artifact of the staining. Maybe these cells just have background level of GIF staining. So we decided to look more closely and co-stain with a parietal cell marker and do some electron microscopy to see what's really going on. And so here you can see staining for ezrin, which is another parietal cell marker in green. You can see staining for GIF in red. And in the control, you can see that the parietal cells are green and the chief cells have these granules of GIF. So you do not see co-staining between GIF and ezrin. But in our knockouts, you could see that these cells that were staining positive for ezrin also had these granules of GIF. You can see it here, some cells more than others. Really excitingly, but not the best EM. So anyone out there who's an EM expert, this needs to be, this is very new data. This is what we are using. There was the data to drive our new R01, but this EM is not perfect. But one cool thing we saw was that at EM, we saw these cells that really looked like a hybrid cell of a parietal cell and a chief cell. And so in addition to having these granules, so the chief cell would have these apical granules to secrete GIF, to secrete the enzymes to digest food, we saw markers of this, you can't really see it here very well, but this membrane, this tubular vesicular network. So the tubular vesicular network is a really important feature of parietal cells that is activated to increase the surface area for acid secretion. And so you could start to see that. We saw a lot of ER in mitochondria. So there's a lot of mitochondria in parietal cells to really drive acid production. We saw that this cell that looked more like a chief cell in the base of the gland actually had a more central nucleus. Normally a chief cell has a basal nucleus and a parietal cell has a more central nucleus. So this was really interesting because at least from the staining data and from the EM, it seems like in the mice where GATA4 is deleted in the parietal cell, there's like a confusion that happens. And so you can see a parietal cell, chief cell, hybrid cell emerging. We don't know whether this is a parietal cell that undergoes abnormal differentiation and gets confused and takes on a chief cell morphology, or is there something about that whole process? Because clearly we know when you lose parietal cells, you affect the chief cells and they can undergo metaplasia. So is it really an indirect effect of losing GATA4? And so there's lots and lots and lots of fun things we're going to do here. Postdocs, come to my lab. There's some of the things we're going to do. We're going to use cut and tag from the parietal cell specifically. So we're going to FACS isolate out GFP positive. So we have a ROSA reporter in there. So we can mark with green with GFP the parietal cells and then do a GATA4 cut and tag. So one caveat to that ChIP-seq data I had showed you earlier was, of course, we didn't have the PCs. So now we're going to be able to do that. So that's exciting. And we can look to see whether or not the same things we saw in the overall bulk ChIP-seq hold through in the cut and tag. We're working on single cell seq because we want to understand what's happening to that parietal cell population, but we also want to see how that's affecting all of the epithelial cells in the stomach. And we're working on RIME, which is a process to basically identify GATA4 binding partners. We're fortunate now that we're able to get specimens from bariatric surgery, so we can do some of these experiments in human. And I'm really excited about the RIME because I really want to understand what GATA4 binds to in parietal cells. That experiment would not have been possible in the mouse because to isolate the number of parietal cells we would need to do RIME, we would need, I don't know how many mice and how many years to do that. So the good news is from the human tissue, we'll have plenty of tissue where we can then isolate using a marker of parietal cells, similar to what Jason Mills used back in his paper in 2000, to pull out the parietal cells and now do, for example, cut and tag in the human parietal cells, do RIME, and we can do single cell seq on the human stomach as well as just bulk RNA seq on those parietal cell populations. We're also working on some injury models. We want to look to see if we challenge them with an injury like a tamoxifen to really delete the cells, will that affect the recovery? We're using Helicobacter because we know that Helicobacter is associated with chronic atrophic gastritis and production and SPEM and cancer. So we want to know how these knockout animals react to that. And we're also doing some cool things with our knock-in animals to look if we can do any rescues and look to see whether or not if we can drive GATA4 from that allele, that ROSA promoter, so it won't be affected by, say, for example, Helicobacter, which we think silences GATA4, pathway downstream of Helicobacter silences endogenous GATA4. If we can put that back in the ROSA locus, which isn't controlled in the same way, it doesn't, as the endogenous gene, will we rescue? So that's some of the ideas we're working on. I threw this in for the Wells Lab, for the Wells Lab and the old, or I should say the Wells Lab alumna in the audience. We're also working on some iPS cell differentiation so that we can examine GATA4's role in development, in human development. So using the iPS model as a model for human development. And we're designing a cell line that we can specifically knock GATA4 out in parietal cells. So use basically engineering an ATP4B cree and an conditional allele that we can then inducibly with tamoxifen, that's what it is, tamoxifen, we can induce loss of GATA4 at different time points. And so with that, that's really the summary of what we've been doing in the lab, studying the esophagus and the stomach, studying developmental biology, trying to apply it to disease, and really the things I love, which are the nitty-gritty transcription factor biology. What does it bind to and who are its partners? I really want to answer the question, why does GATA activate gene 1 and repress gene 2? And so I'm hoping some of that study with looking at the binding partners and then using the sequence surrounding, so using a bioinformatics approach to say when GATA is in a gene that it represses, what factors might bind there? And then in the parietal cell, or even in another cell, does GATA interact with that protein? So that we can set up mechanistic networks and then test those pieces. So all of this, I need to thank all the people in the lab and all our collaborators, and I will take questions. Thank you.
That was awesome. Questions for Michelle?
Yes.
I was curious, so I think this was about midway through the presentation that you were bringing up the different cancer biopsies and you
Yes.
were testing for the different markers. And I was wondering, in the tumor samples that had no expression of the GATA, was there a difference or was there like further investigation as to why?
Yes, so that's it. That's something we're really interested in. At a first level glance, there didn't seem to be a correlation between, say, the stage of the tumor or if it was a more differentiated tumor versus another, but our N is really low. So we might have been able to look at like 15 or 20 so far. So clearly we need to increase that. But I would imagine there must be something like that there is going to be a correlation about something about the type of the cancer or the pathway through which the cancer emerges that's going to implicate GATA4. But I don't have the answer to that, but that's a fantastic question.
From your staining of GATA4 in the control E18 stomach,
Yes.
it kind of looks like there's a little bit of GATA4 that extends into the squamous epithelium of the esophagus, just a little bit. So what is your thought about a transition zone population?
Very good question. Yeah, so clearly that's been something that's been implicated, right? John Wen Q's lab at Columbia has a nice work identifying what they would call a transitional cell that is keratin 7 positive. And when he puts CDX2 in those cells, he can drive metaplasia. We are starting to look at that now. That wasn't something we noticed immediately, and it was something we noticed later. And actually reviewers didn't notice that, so it didn't come up in the paper. But you're right, it does look like there's a few cells there that are at that junction. So we want to now look at those transitional cells, staining with, for example, keratin 7 to see whether those cells match with what Jung-Wang Q said about a transitional cell. And the interesting part about those transitional cells is the idea is they can either become columnar or squamous. And so that in disease, like when you have GERD and damage and reflux, these cells somehow, so there's a lot of controversy about where Barrett's comes from, but that these cells can be the start of Barrett's, that they can migrate in, but then they don't form squamous cells. They turn to columnar. They turn to columnar cells and make metaplasia. So I think it's possible, and we just don't have the data.
You've got GATA4 acting as an activator and or repressor depending on the context.
Exactly.
So it's like, it takes one extra hit to switch in that zone.
Yeah, I'd really love to understand then how,
Yeah.
is that just like a stochastic thing, or is there really something that could be driving that? Yeah. I don't know the answer, but very, very perceptive. Good eyes. Yes,
That was an amazing talk. I have about 15 questions, but I'll stick with one.
okay,
For the parietal cell story,
yes.
do you think you know where the dysfunction is? I was kind of surprised that you even showed canalicula when you're saying that they're smaller, they don't seem to secrete acid.
We didn't get to that
Okay,
yet.
do
So
they
no.
have ATPase?
Yeah, so no, that you're everything you just said is great, and it's all on the agenda of things to do. So no, we haven't done that yet. So the only, like I said, the only data, we have a VM, I should go back and I can pull out and look at that. I didn't look at that completely. So yeah, we can look at the EM and look at what the parietal cells look like in that EM and see whether we see disruption of the canaliculus. Yeah, I don't know of I don't know the answer to that, no. I know that the stomach is less acidic, so it suggests that there's less secretion, but ATP4A and B are still there. So the pump genes are still expressed, but for some reason they're not working, and who knows what that means. Are they being targeted appropriately? Yeah, there's a lot to be unpacked there.
One more quick question.
Okay.
Is the increased proliferation still at the isthmus? or
It
does
is,
go down
it is.
through the-
It actually is it is where you'd expect it to be in that isthmus
So
region.
then you see stem-like cells up through the parietal cell region, but they're not proliferative.
I don't know that so we haven't done staining where we've done EDU with stem markers. So I wouldn't expect them to be, but so we've done EDU staining, we've done stem staining, we haven't combined the two, so.
Good, thank you.
Question from Zoom
Yeah.
from Shazad Sheikh. Great talk, Michelle. Sorry I missed meeting you in person. Have you interrogated TCGA for GATA4 expression and how did acid suppression impact its regulation and binding? It may point to dysplastic lesions that break through suppression of BE to LGD to HGD and adeno cancer.
Can we
Thanks.
copy that somewhere so I get that?
I read it.
Thank you.
I read through and make sure they were all words I could.
No, the answer is no, but yes, now we will. I don't know. The answer is no, we haven't done that, but let's copy and paste that important question so I can go do it, and then we can come back and talk about it later.
I'm emailing it to you now.
Thank you, thank you. The beauty of Zoom there, right, is that the
Yes.
question's all written out, so I can go back and look at it later.
Any other?
Kind of, okay. For the first part of the talk, then, I'll get away from the mature stomach. One thing that I noted like early on is that in the stomach, the units invaginate into the submucosa. Yet when you turn it into a stratified stomach, you no longer get the invagination. So do you think you might be affecting the submucosa or looking at a mechanism of how that happens?
So when we looked, so all the stuff I showed you of the knock-in was at a very early stage at day 14 and a half. So we haven't looked later to see whether, am I understanding your question right? That why don't we see the columnar epithelium forming glands? Did I understand that right?
And kind of the inverse in the stomach, when you have the picture, you have glands and then like the squamous in the middle, and that it just doesn't, there's just changes of whether it's invaginating or not. And I'm wondering if there are changes to the submucosa itself.
We haven't looked at that in depth. One thing we are doing now though, because of course that study and the knock-in, we only looked at 14 and a half. We are like going forward in development as far as we can. We can probably get to about 18 and a half. And then, they have neural tube defects, which makes it challenging because that was one question that was asked of us by a reviewer of the paper is that phenotype, if you take it further, does it actually form a nascent glandular structure? We hadn't done it because it was a little bit challenging to get embryos. So we want to look at that. It would be interesting in both parts. If it happens, great. But Wouldn't it be interesting if you start to have that columnar programming, but then something rescues it and it can form a stratified squamous epithelium again? What would happen there, and what's overriding GATA4? And that could be really cool to think about in disease. So I don't have the answer to those studies yet because poor Jess in my lab has been working really hard. The litter sizes that she dissects are like 3 embryos, and half the time the ones that are the GATA knock-ins are dead, and you can't use the tissue, so it's a little bit challenging for that. The squamous, in the other question about the squamous, so when the knockout, when we have squamous epithelium, that doesn't usually invaginate into the submucosa. It doesn't make that glandular effect, but honestly, I can't say that we did any in-depth characterization of the submucosa, so I can't say no. Yeah.
So I have like 30 questions, but I'll ask 2.
Okay.
So it seems like the GATA4 is kind of this master regulator of this, columnar versus squamous sort of switch, but you need more to get cancer.
Yes.
So when you looked at your single cell transcriptomic data, you did see this like upregulation or enrichment of cancer genes.
Yes.
Did you look at the ratio of cells that had those versus ones that didn't?
No, but we should and we could, yes. We have to really dig into that more deeply, because right now what I showed you, I feel like was just a very almost like you could imagine like a bulk RNA-seq approach, right? Where you're pooling together different tumor regions and saying what's up. So exactly that. We now need to dig into that. And what I wanted to do, and I don't know if this is possible because I'm not an expert of this bioinformatics, but I wanted her to isolate the various tumor regions and then subcluster the cells based on, not just cluster everything across the whole entire tissue, but subcluster so that we could better understand what's going on in those tumor regions, and then maybe get an idea when we can look at, you can't get to the single cell level, of course, with spatial, you get to the couple cell level, but then compare the regions near each other and see exactly that, like what pathways are being upregulated in regions that are looking more squamous, some that are looking more like adenocarcinoma. What about in the regions that have GATA but aren't turning into tumor? And then there are some regions, because EDL2/3 is not perfect, regions that don't express GATA. So we can look at the esophageal cells that don't express GATA and make some comparisons.
So do you think that in those cases that where you have the tumors,
Yes.
do you think that you're getting de novo DNA mutations that are causing that, or do you think it's a microenvironmental effect where you got GATA4 transition, but then you got something else that's not DNA mediated?
Yeah, that's a good question. I don't know that. There is some data out there that we have a very interesting, well, I guess we have this interesting piece of data. When we put GATA4 into esophageal cells in culture and overexpress it for 5 or 10 days, one thing really interesting you see, which I didn't expect, was you see staining for DNA damage. You see gamma H2AX. You see almost every cell expressing that. So GATA could have some role in DNA damage. I don't know that though. That's just me going out on a limb there. I don't know the answer to that question, but clearly would be interesting to look at.
That could hint that you are getting de novo mutations.
Yes.
If you have some sort of impairment of DNA repair, or even just a proliferation, right? Or you might actually get more mutations that occur in a shorter period of time.
It would be also interesting to understand the nature of the tumors in terms of being clonal or multiclonal, and where are the tumors coming? Does if you could go back and look and lineage trace and say, can a cell wouldn't it be cool to know if a single cell then could make a squamous cell or a adenocarcinoma, or there's something specific that happens in a GATA4 cell, like a certain mutation that happens in one, and then that makes it be a squamous cell cancer, or something else happens here and makes it have to be an adenocarcinoma. We could try to do what Sydney Schaefer has been doing at Penn, where she's combining single cell seq with mitochondrial DNA sequencing and allowing a lineage tracing kind of method. We can't do it. So that might be interesting to do, because then you could look at the mitochondrial signatures and try to map the origins of the cells. And then of course, we could clearly look at DNA repair and DNA damage responses and that sort of thing. Yeah. Really interesting question.
Anything else?